Hi guys, welcome to today's episode on cell division and specialization exam questions. I'm going to show you from start to finish how to think, approach and answer the questions to maximise the amount of marks you get. This topic right here is it's not loved by everyone, let's just say that. If you like this topic, then pat yourself on the back because it has difficult concepts to get your head around, especially when questions like this that you can see on the screen really test your knowledge, don't they? So yeah, if you like it, that's good. If you don't like it, hopefully from these videos you'll start to understand that it's okay. You know, the exam questions are alright. You just need to ensure that your content is tip top. Because for this specific topic here, I would say out of the whole specification, you need to memorize every single thing. You need to do it for all the topics, really, but for this one specifically, because if you don't know, they will catch you out so well. It's unbelievable. So yeah, enough scaring you. Anyways, let's begin. So we'll read the question first. It says, suggest an abnormality the scientists might observe in the cells of this tissue. Okay, so from reading that, I don't know what on earth is going on, so I will read the information now. So it says a eukaryotic cell cycle is regulated by 3 checkpoints. Mutations can occur in genes that control the cell cycle checkpoints. Scientists recorded observations of 2 different tissues. In one tissue, the scientists found a genetic mutation that stopped the metaphase checkpoint from working. Okay, so how I would approach this is that I'd ask myself, well, where is the metaphase checkpoint? And luckily for you, I have a visual aid. Metaphase checkpoint is here. Now, if there's a mutation and it stopped it from working, meaning it hasn't even gone, it's basically the stage we're at right now is before the metaphase checkpoint. It hasn't occurred yet, okay? And what is metaphase? Well, when they meet in the middle, okay? A line on the equator. So now it's saying, well, what's wrong, you know? When a scientist looks at those cells, what has gone wrong? Well, there are many things you can say, you know? You can say that the cells contain different number of chromosomes. That's a typical answer, to be fair, whenever you get suggested abnormality. Because when you look at what happens before metaphase, the number of chromosomes matter. Because the thing is, if they are different, okay, most likely caused due to a genetic mutation, then you can't proceed, right? Because it's wrong. Because the cells contain incorrect number of chromosomes. And so that's what I would say here. If you couldn't get the answer, that's alright, because once you know it now, you won't forget it. Because this comes up quite frequently. Let's just say that. Anywho, let's go on to the next question. Let's put this over there. Okay. It says, suggest which cell cycle checkpoint is no longer working in this tissue and justify your answer. Okay, again, I don't know what is happening, so I'll read the information. In the other tissue, the scientists observed cells with chromosomes that had been replicated despite containing damaged DNA. Now, let's go back to the question. It said, which checkpoint doesn't work? Well, what controls replication, essentially? So if I actually bring this back... Oh, that's perfect, great, okay. So, what controls replication? So if they've been replicated despite containing damaged DNA, do you think it would happen here? Do you think we're in G2 stage? We can't be. We have to be in G1 stage. And this might be a bit confusing. It says it's been replicated despite containing damaged DNA. So that means you need to work backwards, not forwards. It'll be G1 because it's allowed for it to continue. And in S phase, the DNA synthesis, it's been replicated, so it can't be G2 because it had to happen before it. And so thus you would say it happens in G1 checkpoint because, I mean, we have damaged DNA here. So that's justified. And the reason why is because, in the G1 checkpoint, cells with damaged DNA should be stopped from entering S phase. But it didn't. So that's why it's G1 checkpoint. Okay, so this one seems like a very simple question, just state all the letters. But actually it trips up so many different people, okay? Because let's read it. State all the letters in the figure that represents the phases of interphase. Phases, okay? Not checkpoint, phases. So here, what would be the phases is G1, S, and G2. That's it. The reason why I put this question in the video is because if you said M or C, you don't get the mark, despite saying G1, S, and G2. So this is why you need to be careful what you write. Be careful of two things. One, saying more than you need, and by having something that you wrote part of the do not credit part of the mark scheme. And number two, contradicting yourself, because even if everything you wrote is correct, but if you contradict yourself, that's it, the mark's gone. Okay? So you need to be wary of that. Because again, actually think about this like psychologically here. Notice how phases is in bold. It's because people tend to basically trip up on this. They start talking about checkpoints or including checkpoints. If the exam question has clearly put something in bold, read that part over and over again until you realise why they've done it. Because I'm telling you, they don't put things in bold without a reason. So yeah, that's why here would be G1, S, and G2. Okay? Anywho, and that is the end of today's episode. Remember, the two most important exam techniques are read the question first and state all the obvious points. Now over to you. Try some questions and let me know how it goes.